We present a technique for manipulation of giant vesicles using localized calcium ion gradients. The main advantage of this technique is the entirely contactless stimulation of the vesicle surface and direct visualization of lipid membrane remodeling upon interaction with calcium ions.
Our approach introduces a means to address the details of calcium ion membrane interactions, providing new venues to study the mechanisms of cell membrane reshaping upon changes in the cell microenvironment.
To begin this procedure, prepare a solution of lipids in chloroform. The final concentration of lipids in chloroform is 1 mg per milliliter, and the final mass is 0.6 milligrams. Wrap the glass vial containing the lipid mixture with metal foil to protect the contents from ambient light. Place the metal foil-wrapped sample vial inside a glass beaker. To remove the chloroform, evaporate the solvent in a rotary evaporator for 3 hours with the pressure reaching 7 kilopascals vacuum at 80 rpm. A dry lipid film is formed at the bottom of the glass vial after evaporation. Slowly add 600 microliters of phosphate-buffered saline solution on top of the dried lipid film. Following the addition of the buffer, gently add 6 microliters of glycerol to protect the sample from complete dehydration during the formation of a giant unilamellar vesicle connected to a multilamellar vesicle, or JoVE MLV. Seal the glass vial using parafilm,